Hi, this is Mr. Andersen, and I'm back for another lesson on mitosis, as it is one of the smallest steps in the cell cycle. In the first step, a nuclear envelope encloses the nucleus and two centrosomes have formed during the S phase of interphase. After the first step of mitosis, prophase goes on its way. Now, the centrosomes move away from each other, and the spindle also begins to form, while the mitotic spindle also begins to form. The chromatin fibers become more tightly coiled and start looking somewhat like chromosomes. The nucleoli also disappears, and the duplicated chromosomes appear as two identical sister chromatids. Also, the nuclear envelope starts to fragment. The next step, or prometaphase, is one of the phases people don't really cover that much. But it needs to be addressed, as the centrosomes move to the opposite sides of the cell, more spindle fibers form. The nuclear envelope also begins to disappear even more than before. Now, as you can see, the chromosomes are becoming even more dense than they were, and they now look like actually paired sister chromosomes. The more spindle fibers that are being formed are called kinetochores as they connect to the centromere. Okay, so we're just gonna wait a couple seconds until she's done erasing everything and starts up again. Okay, now that that's over, metaphase is the next stage in mitosis. Everyone usually knows this phase because the chromosomes always line up at the middle of the cell, known as the metaphase plate. As the centrosomes are on opposite poles of the cell, the kinetochores of the sister chromatids are attached to the kinetochore microtubules coming from the opposite poles. Now, let's talk about anaphase, as it's the shortest stage of mitosis. When the cohesin proteins are cleaved, it allows for the sister chromatids to part suddenly, as you can see here. The two liberated daughter chromosomes start to part toward opposite ends as the kinetochores begin to shorten. By the end of anaphase, the two ends of the cell have equivalent and complete collections of chromosomes. The last steps are telophase and cytokinesis. In telophase, two daughter nuclei begin to form, nuclear envelopes arise from parent cell fragments, nucleoli appear, chromosomes become less condensed, and any remaining spindle microtubules become depolymerized. As mitosis is finally complete, cytokinesis is the division of cytoplasm as it involves the formation of a cleavage furrow in animal cells, as it pinches the cell in two, but in plant cells a cell plate forms, forming a new cell wall. Thank you. I hope you learned a lot about mitosis, and I hope that's helpful.